Hi, everyone who's joining us right now. Good evening. Thank you for joining us this week for the HEALEY ALS Platform Trial update. We'll jump right in just a moment as we let everybody in from the waiting room. My name is Allison, and I work in the patient navigation area for the HEALEY Center at Mass General. I'm joined today by my partner and my teammate, Catherine Small, from the patient navigation area. I'm so excited to welcome Dr. Lori Chibnik back from Harvard School of Public Health and MGH to talk to us today about statistics and placebos. So as everybody is logging in, I'll go ahead and turn it over to Catherine to start.
Sure, so we can go on to the next slide. And as we do every week, just want to provide a quick update on how things are going with the platform trial. So for those who are returning, this is old news, but for those who are new, welcome. We currently have completed enrollment for the first 4 regimens, A, B, C, and D. But Regimen E, the latest regimen, is open to enrollment at teaser trailer. There's going to be 41 centers on a slide and a couple slides, but we have 41 centers that are actively enrolling participants for Regimen E, which is testing an experimental drug called trehalose. We can go to the next slide. To date, 96 individuals have signed informed consent, so that's at the master protocol level. And for Regimen E specifically, there are 66 people who have been randomized. So that means we're at 66 out of a target of 160 people who have entered Regimen E of the trial. And as always, we just want to say thank you to everyone who has participated, expressed interest in participating, or serves as a caregiver or family member supporting someone in the trial. It takes a village, so thank you. And we can go to the next slide. Here is that 41 sites that I was talking about. So I think since last week, we added a couple, Augusta University in Georgia and the University of Utah, both of which are brand new to the platform trial. So we designate them in green tags on the map there. So big welcome to those 2 sites. And they are now actively enrolling participants for Regimen E. If you use the QR code there, you can go to our website to see the site map and the contact information if you wanted to reach out to a site. I think one more slide here. And here, again, some QR codes that are handy if you wanted to register for future weekly webinars or sign up for the ALS link, which is an email information broadcast system for ALS research and news. Upcoming guest speakers to be determined for the beginning of August, but on the 18th, we'll have Dr. Peltier from Vanderbilt University joining us. All right, and without further ado, this next slide I'm particularly excited about because we have Dr. Chibnik back as our guest speaker of honor and our go-to biostatistician for all our questions regarding the trial, and this week especially regarding placebos. So I'll hand it off to you, Lori.
Oh, thank you. Thank you very much, both Catherine and Allison. It is a pleasure to be back. So hello again to those who have seen me before, and hello for the first time to everybody else. The plan is I'll talk for maybe 10 minutes about placebos and how we as statisticians think about placebos. And then we're going to open it up for Q&A. There is a place for you to type in your Qs, and they pop up on my screen for me to give the As. And so we should have enough time to get through most of those. And I'm also, if there's anything I can't answer right now, we write it down and we follow up later with sometimes a little overly detailed answer, but we try our best. So I'm going to start the way I did the other biostat seminars and introduce myself. My name is Lori Chibnik, and I'm a biostatistician. I have been for a significant amount of my life, I'm told. And I really love what I do. I work with the ALS trial here. In fact, instead of staring at my face, can we click to the next slide? I am one of a team of biostatisticians. We have actually now 6 people at MGH Biostats. That will be updated. We have somebody that just started very, very recently, another master's level statistician. So we are a large team of biostatisticians working together on this trial. We also work with another team of biostatisticians at Berry Consultants. So I had started this whole webinar series to give you guys an introduction on how biostatisticians think. I can't speak for all of them, but I will say in general, one of the things that I always like to talk about at the beginning of this is that as a biostatistician, we're trained to think about groups of people. Our entire career, everything as we put together and as we think about these trials and as we think about analyses, is this group of people different from this group of people? And that's just how statistics work, because we want to be able to look at this data and say, based on what we see in our sample, we think that this is what is happening in the population of people. And I realize, especially as I think about talking about these webinars, is that it really is a privilege that we have as statisticians to be able to think about a group of people, and that many people in the audience today don't have that privilege of thinking about a group. They're thinking about a one, and that might be themselves, or that might be a loved one who is afflicted with ALS. And I realize that my privilege of being able to think about this group is very different than the listening of really being concerned about that one individual. So what I ask of you is to come halfway with me and understand that what we're going to talk about is how we think about it in the trial, in the larger population of the ALS community. And in turn, I'm going to come also halfway and realize that everything I say is, while meaningful, might not be the most meaningful thing that someone who's thinking of n of equal to 1 might be thinking. So again, this is something about, just trying to understand where each of us are coming from. And on that note, can you go to the next slide? When I look over here, it's because that's where all the slides are. So today's topic is placebos. And I put a lot of thought into this one. I was a little afraid to venture down this road, but I'm going to talk a little bit today. Next slide, please. About placebos. And I broke this down into 5 areas. First, what is a placebo? Second, why do we need them? Third, what is this thing called a placebo effect? And then specifically, what is a placebo effect in the context of ALS? And finally, a question that gets asked quite a bit is, can we use placebos from previous trials? Okay, so next slide, please. So very quickly, what is a placebo? I have this written up here as a sugar pill. That's a terminology that a lot of people use. It's misleading, because what a placebo is it is a pill, a drug that is identical to the actual drug, with one exception, and that is that active ingredient. So it's not made of sugar. It is identical in look, weight, taste, smell, dose, any property you can think of to that active drug, except it doesn't have that ingredient. And as I started learning more about clinical trials in my training, I found out that the area of creating a placebo is like a whole area of science, because not only does it look and weight and taste everything the same, but if you were to take a pill and a placebo and drop them in water, they would dissolve identically. Like if you were to drop them on the floor, they would bounce identically. They have to be perfectly identical. And the only way to tell the difference is through some sort of chemical analysis. So it's not really a sugar pill, and it's not necessarily always a pill, of course, but it's as identical. And the only way to tell the difference, again, is through a chemical analysis. So next slide, please. So why do we need them? So the ultimate goal of a clinical trial is to show that a drug is both safe and effective, right? So we need to show that any efficacy, so any benefit that patients would get from this drug, is due only to that drug and that drug alone. And also that any safety issues, so any adverse events, any side effects, any safety issues that we might see in the trial, is that due only to that drug and that drug alone, or could it be due to something else, right? So this design of this double-blind clinical trial, which is what the HEALEY trial is, where neither the participant nor the researcher knows the drug that someone's getting, is the best way that we have. It's the gold standard to try to determine this. So I say here that neither the participant or the researcher. It actually goes further than that. For this particular study, the participant doesn't know, the researcher doesn't know, the pharmacist doesn't know. So your local pharmacist that hands the drug to your nurse or your doctor or whoever is giving you the drug They don't know. The woman or the man who is administering the IV, they don't know. The only people that know is a central warehouse where all these drugs are labeled with just numbers, and then there is a list somewhere that identifies those numbers as the drug or the not. So then they're told, pick up the drug that has this number on it and hand it to that particular participant. So nobody knows which one is which. Okay, next slide, please. Thanks. So I'm not sure if this is a new phrase, but the placebo effect, it's something that's spoken about quite a bit. So what is the placebo effect? It is a beneficial health outcome resulting from that person's anticipation that this intervention will help. So that's one way of thinking about it. It's really a beneficial outcome resulting from being part of this study or taking this drug or being observed by a doctor. We tend to think of the placebo effect as seen more in subjective measures as opposed to objective measures. So subjective measures is how long did it take for your headache to go away? Whereas objective measures are things like taking a blood test and measuring some blood marker, right? But that isn't actually always the case. It isn't always subjective versus objective, which we'll get into in the next slide. But there are some interesting examples of studies that have been done on placebos. There's a whole area of work on the placebo effect. And people that have done studies on higher dose placebos being more effective than lower dose placebos. So the participant might be told that this is 20 milligrams and that one is 10 milligrams. And knowing that you got the higher dose, we see a higher placebo effect. The same is true for more expensive placebos. So participants being told this one costs $10 a pill and that one costs $1 a pill, actually showing placebo effects. So there's a lot of very interesting work being done on placebos. Next slide, please. But specifically, what is a placebo effect in a disease like ALS? So ALS progression has very little subjectivity. However, we do see placebo effects in some of these measures that we might think of as being more objective. So some examples that I saw when I looked more into the literature just specific for ALS is that there was an open label trial of lithium. Open label means everyone knows what the drug they're getting in a very small number of patients. And it ended up suggesting that this drug helps slow the disease. So in order to prove that, they did a larger placebo-controlled double-blind trial and found that there was no effect of lithium. So the first study was a small group of people where they knew that they were getting a drug that people thought might be effective. It looked like it slowed the disease, but in the double-blind study, it showed no effect. Same, you can see examples in animal studies, and many of these drugs go through animal trials before they go into human trials. So in animal studies and this open label, so people know what they're getting, human trials suggested that minocycline was beneficial, but again, the larger placebo-controlled trials showed that it was not, and it even showed a little bit of harm. So we do see placebo effects in ALS. They're slightly less, but they do actually exist. Okay, so next slide, please. So a question that we get quite a bit is, can we use placebos from previous trials? So we've had some previous trials in ALS. There were placebos in there. Why can't we just use the data from those previous trials? So the issue is that, remember, we need to show that any efficacy and adverse events are the result of this drug that they are giving in this trial and this drug only. So any differences between a current trial and a previous trial are going to cause that to be a little bit more cloudy. We're not really going to be confident that it was the drug that caused the benefit and not just the way this new trial is being run. Any differences. So maybe the previous trial followed people for a shorter amount of time. Maybe the previous trial had different inclusion and exclusion criteria. So maybe it was only people with a shorter disease duration or longer disease duration. So any of these differences can cause a difference in our current results if we compare treatment to a previous placebo. And that there's been a lot of work on that in terms of statistical work that shows that by adding previous placebos, you actually add more noise to your data. So if anyone has heard the phrase signal over noise, we always try to find our signal over the noise. So we try to keep that noise really little. And if we use other placebos, we're bringing more noise. So basically any noise that was in a previous study, we're bringing into our new study. It makes it harder to see the signal. And they've showed that this is true with what's called simulation studies, where we basically create a fake study with just data and show what we can change within that simulation that causes the data to not find a difference when there really is one. So some of the other changes are changes in the standard of care. If there's a new drug on the market, and edaravone is one of the examples, that people might not have been taking previously, but more and more people are taking now, those placebos would not, the previous placebos would not be an appropriate comparison with any sort of new group, new study. Okay, so I think one more slide. Yeah, so those were some of the questions that had come in before. So those were the ones I wanted to answer. I also just wanted to talk a bit about placebos. So with that, I'm going to stop talking and I'm going to go look in a slightly different direction with some of the questions that came up. So I have one question. I don't agree with the position that the ALS progression has little subjectivity. The typical primary endpoint of ALS-FRS has most questions that have a rather high level of personal assessment and subjectivity, such as handwriting, dressing difficulty, gait disturbance, and speech impact. That's correct. And thank you for pointing that out. That is very correct. ALS-FRS is many, many components, and a lot of them have some level of subjectivity to it. The way we try to limit the subjectivity as much as possible is through training and training and training. So the clinicians that administer the ALS-FRS, anyone who administers the ALS-FRS has to train and train to try to make sure to get rid of as much subjectivity as possible. So that's one thing. The second thing is that we're not really comparing this group's average ALS-FRS to this group's average ALS-FRS. What we're comparing is the change in ALS-FRS in this group to the change in ALS-FRS in this group. And I probably could have said it a slightly different way, which is we take everybody's individual change. So each participant is compared to their own previous ALS-FRS. So their own trajectory of ALS-FRS. And then we average those trajectories. It's not a simple slope, although y equals mx plus b does factor into the equation in one way or another. We actually are able to take the slope and take into account that it might not be linear in some of our analyses, but not all of our analyses, but some. And so you're compared to your own. So this group that had the drug, was their progression different than the group, the placebos, those combined placebos that did not have the drug? So that is another way to try to take out subjectivity, because you can argue that your own subjectivity shouldn't necessarily change over time. Not going to say that that's absolutely the case, but it shouldn't necessarily change over time. And then there are some objective measures, hand strength, SVC, vital capacity. There are some actual objective measures that are secondary outcomes, so not our primary analysis, but our secondary analysis, which are also somewhat less subjective. Although I've heard the argument of I just had a bad day. So there is a little bit of that factors in as well. Okay. Why is it okay not to have placebos in oncology clinical trials, but we need them for ALS clinical trials? That's an excellent question. So what they do in oncology clinical trials, and this is a question that comes up whenever I teach about clinical trials with entry-level biostatistician grad students, is when can you have placebos and when can you not? So with oncology trials, since for many of those diseases there is a standard of care, you don't have a placebo. What you have is you have a group that gets the intervention and a group that gets the standard of care. So they're still getting something. They're just not necessarily getting the intervention. There could, and for many of those studies as well, since it's unethical to not give a cancer patient the known standard of care. So this is a disease that might have a known chemotherapy regimen. You will then have to give your new drug on top of the standard of care. So many of the oncology clinical trials, everybody in the trial gets a standard of care, and double-blind portion is that some of them on top of it will also have this additional treatment, this additional compound, and others will get a placebo on top of the standard of care. This is not different than what we're doing with the HEALEY ALS. Every participant gets the standard of care that is given by their clinician. That's why many participants are on some of the already prescribed medications. But it seems a little more obvious than the oncology trials that like they don't talk about placebos as much in the oncology trials necessarily. But that's how a lot of them go. I'm also speaking about phase 2 and phase 3 trials. So the HEALEY ALS is a phase 2-ish trial. It falls more under the phase 2. Phase 1 trials, there are no placebos. So phase 1 trials are everybody gets a drug. It's the first time it's really given in humans. We want to see what the tolerance is. We want to see how the body reacts to it. And those are phase 1 trials. They're much, much smaller. They're much more much more restrictive on who ends up enrolling in those studies. And that's a slightly different thing. So there are a lot of oncology phase 1 trials as well. I will say that also brings up one other, I'm noticing the time and I have a couple more questions, which I promised to get to. They told me I can go a little later. Yes. Somebody asked the question that I answered at the exact time I answered the question. It is true that for phase 1 trials across all diseases, that all sick folks get the true drug. That's what a phase 1 trial is. Yes. Absolutely. It's to prove that the drug is tolerable in humans and is safe and effective and shows the same as what they saw in model organisms, which is a fancy way of saying animal studies. But I was going to say, I forgot what I was going to say, but it will come back to me. Oh, yes. So the standard of care. So if one of these regimens turn out to be effective, and we won't know until we get the results, that might become the new standard of care, which means moving forward, everybody in the study might be on this new drug, and then we're going to test another drug on top of this new drug, which makes it even harder to use previous placebos. Every ALS patient progresses at a different rate. Does this add to the complexity in determining if a placebo versus an actual drug works? Yes, it does. ALS is a very complex disease, so it does add to the complexity. We try to control for that by looking at their pre-baseline progression. So before they got the first dose of drug, how did their ALS progress before they started compared to after they started? So we can see if your normal rate of progression was like this, but then you took the drug and it got better. That shows us that you're improving based on yourself, even if it doesn't necessarily look slowing down in the study itself. So we do try to take that, we use that as part of our model. Pre-baseline slope is usually what we call it, to try to control for the fact that everybody progresses at a slightly different rate. In case the study drug is active, how do you reduce the disadvantage of placebo? I have to apologize because I'm not sure exactly what that question is asking, but I wanted to read it out loud. And if that was your question, can you throw it in again to the Q&A and I'll hit it? In case the study drug is active, how do you reduce the disadvantage of placebo? Well, I'm going to answer it the way I think you might be asking, but I will keep looking to see if you pop up again. So everybody at the end of the study, regardless of where they were in that double-blind portion, has the option of continuing on an open label extension. So after those 24 weeks, you have the option of going on open label extension, which means for the people that were on active, it just continues. And for the people that were on placebo, they now know that they will be on active. The other option is after a short washout period to go back to the beginning and go into a different regimen and try again in a different regimen. So that's what we do to try to give everybody the advantage of this potentially new effective drug. I should say new potentially effective drug. So if that's how you were asking the question, I hope I answered it. Since regimen A washed out for futility, do you still analyze its effect? Believe that was regimen B. Was it regimen? Let me just say it this way. When a regimen ends early for futility, we do still analyze it, and the placebos in that regimen are still used for analysis with the other regimens. Absolutely. We still analyze the effect. We still look at the data. We still look at the secondary outcomes, et cetera. And I have one last question, I think, yes. For patients who don't qualify for the interventional trials, how can smaller biotech sponsors who might not feel that they have the funds to offer expanded access be convinced to offer compassionate use for that individual patient if they aren't part of HEALEY's EAP? That is not a statistical question, but I am going to try to answer it as a stat. There's another question that popped up. So I'm going to think about how I'm going to answer that question while I answer the other question, and we are going to come back to it. If a drug such as ILP is used in lieu of Realizol, I believe, when you have to run a placebo-less trial, the phase 1 required to wean off of the other drug. If a drug such as ILP is used in lieu of another drug, would you have to run a non-placebo trial, a phase 1 required to wean off of? No, you don't. A phase 1 trial is not always required. It depends on the situation. If it's a brand new drug to humans, then in general, it's going to be required. But I don't necessarily, I'm not sure about this particular situation, so I don't know. I don't know, but I will write that down and see if we can get the answer. Now back to for patients who don't qualify for the interventional trials, how can a smaller biotech sponsor who might not feel that they have the funds to offer expanded efforts be convinced? I'm not sure. I'm not sure. So if they have data and we analyze the data and we find after a small study is over that there is no significant differences, that p-value that we talked about it last time, sometimes they will look at smaller subsets of their data and say, well, let's just look at men, or let's just look at women, or let's just look at this type of ALS, or let's just look at people with short duration, and maybe they'll find something in there, at which point they would then need to do another study, but it would be a convincing analysis to say like, hey, it looks like there might be on, we're not sure, maybe to convince them that it's worth to invest to do a bigger study. In terms of other ways to invest to encourage these smaller biotech sponsors, I think patient advocacy groups are the best way to actually like get in people's ears and explain to them what these drugs really mean. But in terms of a statistician, the best I can do is analyze the data, give them hypotheticals and say, this is your probability based on what we saw. If you do a bigger study, you might see an effect, at which point you have a drug. That's what we as statisticians will do. But we're very honest and we like to do things in numbers and math. So we'll say things like there's a 80% probability if you have this many people and this is what really is happening that you will see an effect. And that's how we as statisticians get to contribute. So lastly is the previous question that apparently I did answer correctly. Thank you. You answered my question. Yay. One additional aspect, interim analyses to make sure to reduce exposure to ineffective treatments. Yes, we do interim analyses. That is the other way. I don't know if you're actually answering your own question, but you did. So the other way to decrease the amount of burden on people who are on placebos is we have these interim analyses. And there is a group, in fact, you saw a picture of them, very consultants who do not work for MGH, do not work for the drug companies, who get every 3 months the data, and they have the real randomization, and they look at it in a very prescribed way that says before they even look at it, if what we see is this number, if the probability that we'll find success is less than a certain percent that is predefined and the FDA agrees to, then we stop the study for futility, because it's unethical to keep people on to keep giving people this drug because we know it's not going to show an effect. I will say that every 3 months, it's called the Data Safety Monitoring Board. They look at the interim analyses to decide, is it futile, we're not going to see an effect? They also ask the question, is the effect so big already that it is unethical for us to not give everybody this drug? So there is another criteria that if it's like so effective that we want to stop it early to give it to everybody, that's a high criteria to match as well. So they can decide that. They also look at safety. So if they see that there's a safety issue with the drug, maybe they would advise stopping early because we feel that it is unethical because there's a safety issue. So interim analysis, exactly. Thank you to add that question, that answer, because that is absolutely correct. And that seems, oh, thank you. There was a nice comment. I thought I wasn't going to understand the webinar, but you explained it. Thank you. I love math. I love statistics. And whenever people find out I'm a statistical professor, they get a look of horror on their face. And I hate that. So I'm sorry, and I apologize for every bad statistics professor you guys might have had in college. I apologize for all of them. So I'm glad. I'm glad that I can share what we do, and we work really, really hard to get this, to make this be a very effective, a very efficient trial, the least amount of burden on patients, and as quick as we can to get these results.